Hello everyone. In this session we are going to take you through what we call lab diagnosis of African trypanosomiasis. Previously we looked at the life cycle. So in this session we want to see how can we confirm the presence of sleeping sickness from a patient who is showing signs and symptoms. Please remember to subscribe if you haven't, like and share to many others. So with African trypanosomiasis, we saw it is caused by two species, majorly T. gambiense and T. brucei rhodesiense, as we saw in the previous video. So in this session, we are going to see how can we confirm the presence of these species, that is T. brucei gambiense and T. brucei rhodesiense. So these are the two major species which are leading to African trypanosomiasis, whereby rhodesiense is common in woodland and savanna, and is also found in the game parks, and it causes what we call East African trypanosomiasis, whereas gambiense causes West African trypanosomiasis. So, what are the specimens we get from the patient to use to diagnose this sleeping sickness? And one of the samples or the specimens we use is blood, whereby we can collect both venous and capillary. All can be used depending on the amount of blood needed and the method. Then another one we can use is CSF, and this cerebrospinal fluid is collected throughout chronic infection or severe chronic which has crossed the CNS. Then another specimen we are going to look at collection of chancre aspirates. We said this chancre is a reddish nodule, painless at some time, which is formed at the site of inoculation when the metacyclic trypomastigotes are injected by the Glossina fly. So at the chancre, we can easily collect there the aspirates. We can even use peritoneal aspirates, that is from the peritoneal cavity. Then we can also use lymph node aspirates. Lymph node aspirates. So these are the type of specimens we can correct to diagnose African trypanosomiasis. So after looking at the different specimens, now we are going to look at the methods we use to diagnose. And as we start the methods, I want to tell you that the infective stage of African trypanosomiasis is what we call trypomastigote. And this trypomastigote is found in the peripheral blood, can be found in the chancre, can be found in the lymph nodes, and at the same time in the central nervous system. That's why we are correcting the CSF. And the features of this trypomastigote, this is the posterior end. So this side is what we call the posterior end of the trypomastigote. This is the posterior end, and this is the anterior part. So it has what we call the flagella, and this flagellum enables it to swim within blood. So it is motile in blood, and that's why we can even use direct wet mount to look for the presence of motile trypanosomes. Then it has what we call the vaulting granules. It has the nucleus, which is at the center. Then it has the axoneme, and the axoneme is with the kinetoplast. So the kinetoplast is found near the posterior end, whereas the nucleus is found in the center, and this trypomastigote is S-shaped, which is different from T. cruzi, which is C-shaped. So whenever you examine a wet mount, you examine a stained film, and you find for the S-shaped trypanosome with a single flagella and a posterior axon nucleus plus the kinetoplast with what we are calling an indurating membrane. So this is an indurating membranes. So these indurating membranes are very prominent, and this one is what we are going to see in films, either using concentration techniques or using direct wet mount or in stained thin film to confirm the presence of African trypanosomiasis. So the first method is the direct wet mount. Here you collect blood or CSF. Here we can use blood or CSF, or even the aspirates, both the chancre and the lymph node aspirates. So after collecting these specimens, either they can be concentrated using triple centrifugation test or microhematocrit buffer coat method or miniature ion exchange centrifugation test. And after concentrating them, why do we concentrate them? We want to increase the chances of finding these trips. So after concentrating them, we can easily examine. So we put a drop of the sample. If this is a clean slide, glass slide, we come and we put a drop of the concentrated whole blood. It can be whole blood, it can be serum, it can be buffer coat, or CSF or aspirate fluid. Then we apply what we call a cover slip. After applying a cover slip, then we examine under microscope. Examine under microscope, either using ×10 and ×40 objective lens. So these are the objective lenses we can use to look for motile trypomastigotes, which we said they are S-shaped. These trypomastigotes are S-shaped. So that is the direct mount method. Another one, after seeing the presence of the motile trypomastigotes, we go ahead and fix. We fix this smear using either absolute methanol. Then after fixing, we stain. It can either be thick film or thin. So both thin and thick film can be made, whereby if I have a microscope slide, this is the frosted end, we can make a thick film, and at the same time we can make a thin film. So this is the thin. So this one we are calling it thick film. This is thin. So all of them are made. For the thick, we do not fix. So don't fix a thick. We only fix the thin film. After fixing, we stain using either Gimusa stain. Gimusa is the most preferred Romanowski stain for demonstrating trips. But if you don't have Gimusa, we can use field stain. Field stain A and B. So these are the ones we can use, or sometimes we can even use Leishman, though in preference we prefer Gimusa. So after staining with Gimusa, we can use either 10%, we could prepare 10% working solution and we stain it for 10 minutes, or we use 30%, we use 3% and we stain it for 30 minutes. So with Jimusa, if we use 10% Jimusa, those are 9 drops of the buffered water plus 1 drop of the Jimusa Kong. That is what we call 10%. You stain for 10 minutes. But if you use 3%, there you stain for 30 minutes, air dry, and then come and examine. So when we are examining a thin film, we are looking a thin or thick. We start with a thick film. And what are we looking for? We prefer thick because it increases the chances of getting the presence of trypanosomes. So we look for the presence of trypomastigotes, as we have seen there, its characteristic here. Or another thing, we look for the presence of morula cells. You can call them morula cells or moat cells. In a thin film, we can also look for the presence of leukocytosis. That is increased white BC count, which is always seen in trypanosomiasis. So how do moat cells look like? In a stained film, the moat cells, if this is a monocyte, you find it has its kidney-shaped nucleus, then with vacuoles, with clear vacuoles. So presence of these clear vacuoles, or which looks, these vacuoles looks like air bubble within a white blood cell. So this is what we are calling a moat cell. A moat cell, or you can call it morula cell, which confirms also presence of trypanosomiasis or trypomastigotes. So after looking at the thick and thin film which are stained with either Jimusa or filled, we can also go ahead and use this blood collected in EDTA to do what we call complete blood count, CBC. So CBC is known as complete blood count, and under complete blood count, we want to rule out conditions like anemia. As trypomastigotes are in the blood saturation, they lead to anemia. That is due to increased destruction of RBCs. So we can do, and we look at hemoglobin levels and total RBC count. It is reduced, leading to anemia. We are looking for presence of leukocytosis. That is increased levels of white blood cell. Then also we are ruling out under CBC. So we are looking for reticulocyte. The reticulocyte count is high because there is increased destruction of red blood cells. So we can see increased reticulocyte, increased white blood cell count, and anemia on a CBC. That is what we can see there. Then if we do cut, that is cut in full, is cut agglutination trypanosome or trypomastigote test. So this cut is looking for the presence of antibodies, which you can call immunoglobulins. So these antibodies, that is IgM and IgG. But we prefer IgM because it is the one that shows current infection. So here you are using an agglutination test or a latex coated with antibodies. No, the latex, if this is the card we are using, this is the card, it is pre-coated. It is pre-coated with what we call trypanosome antigen. So these antigens are going to look for presence of antibodies in a patient's serum. So here the sample we use is serum. Serum is preferred when we are doing antibody tests. Though currently we are using advanced techniques like ELISA, but this cut was originally being used for diagnosis of Trypanosome brucei gambiense, especially when you go to the field where there is large number of patients. You can easily screen them either if they are positive or negative by using this card agglutination trypanosomiasis test, so that you know they are positive or negative. But in case they are positive, you have to go ahead and do these further tests, like the stained thin film, the direct wet prep. You go ahead and after doing these centrifugation techniques, and you're able to confirm. So this antibody test is not confirmatory, so you have to go ahead and confirm, because it is not specific. Very sensitive, but not specific. So that's why we go ahead to do other tests. But we have said currently we are doing other tests like ELISA, that is enzyme-linked immunosorbent assay, which have become more accurate compared to the CAT, which was previously being used. And this is a serology test where you have a kit, then put the reagent containing the antigen, then add. So if we had an antigen, we add a drop of the antigen, that is the reagent containing the antigen. Then you come and add equal volume of the serum, patient serum. Then you mix. After mixing, you look for presence of agglutination. And if you see agglutination, agglutination is seen by clamping of cells at the test site. If you see clamping, you say positive, then you go ahead and do these confirmatory tests. That is what we can talk about. Another technique we are going to look at, from 5 to 7, we are looking at the concentration techniques which we can use. For example, triple centrifugation is a concentration technique that is aiming at concentrating presence of trypanosomes so that we increase the chances of seeing them into peripheral saturation. So this triple, as the name suggests, triple. Triple means triple then centrifugation. It means we centrifuge 3 times, whereby the first time we centrifuge, we collect 9 mils of venous blood, EDT anticoagulated, and we put it into what we call a tube. And after putting these 9 mils into a tube, the first time of centrifugation, we centrifuge at 1,000 revolutions per minute for 10 minutes. This one will give us what we call, then we remove the sediment, we shall remove the spinatant, and we remain with the sediment. Then the second time we centrifuge at 3,000 revolution per minute, still for 10 minutes. After that, here we shall collect the spinatant. The spinatant, here we centrifuged the spinatant, and here after getting this, we are going to centrifuge it further. You collect the spinatant together with the part of the buffer coat, because we see trypanosomes are concentrated more in the buffer coat. So we centrifuge it further here at 5,000 revolutions per minute, still for 10 minutes. So you can see we centrifuge 3 times. You centrifuge as you obtain the the spunetant containing the plasma and the buffer coat. If it is CSF or it is the aspirate, you obtain the clear part. But later, at the end, we don't examine, we examine using the deposit. So at last centrifugation, for 10 minutes, we obtain a deposit and we put it on a slide, on a microscope slide. So we put a drop of the deposit on the microscope slide, that is direct wet mount, put a cover slip and examine using times 10 and times 40. If there is a presence of trypomastigotes seen as S-shaped with flagella when they are motile, then we go ahead and we examine a thick and a thin film and a thin stained film. So this is what we can do after centrifugation of 3 times with increasing speed. As you can see, we started with 1,000, we went to 3,000, we went to 5,000, then at the end we collected a deposit which we were able to use. And its major aim is to concentrate or to increase the chances of seeing trypomastigotes. Then number 6 is microhematocrit buffer coat method. This is the most accurate and sensitive compared to triple. So this triple is done where we cannot obtain a microhematocrit centrifuge. But if you can obtain a microhematocrit centrifuge, this one becomes the best method of concentrating the trypanosomes. And how do we do it? We collect venous blood, or we can collect CSF, or we can collect the aspirates, and then we fill them to 3/4 within the capillary tube. So these are capillary tubes number 1 and number 2. We fill them with blood. After filling 3/4, we fill it 3/4. This one is better run in duplicate to enable balancing of the microhematocrit centrifuge. So after filling both the 2 tubes 3/4, we come and seal one end. So we seal one end using a sealant such as plasticine. After sealing one end, we come and orient it in microhematocrit centrifuge. So we can come and orient one here, orient the tube in this slot when the sealed end is facing outward. So this is the sealed end. We come and orient another, because we filled the 2, you come and orient another one in this, but with the sealed end facing the periphery, because we are using centrifugal force, which is the outward pull. So after balancing, now we set the time for 3 to 5 minutes at a speed. Here we use the speed of 10,000 revolutions per minute, between 10,000 to 12,000 revolutions per minute for 3 to 5 minutes. After that, but before applying the speed, we first seal, we first cover tightly to prevent this capillary tube from coming out of the slot. So you cover, after covering, you set the timer and centrifuge at 10,000 revolutions per minute. After centrifuging, we are going to obtain, if this is the capillary tube, and this capillary tube is sealed at one end with the plasticine, and we have obtained the cells. We know that red cells settle first at the bottom, followed by a clear buffer coat, then plasma. So this is the plasma. So we are interested in this part which we call the buffer coat. That's why the method we call it microhematocrit, because we are using microhematocrit centrifuge. Then buffer coat, because trypanosomes are found in the buffer coat where we find the white blood cell, because their density is the same as for the white blood cells and platelets. So they are concentrated in the buffer coat. So after obtaining this, what you do, this is the microscope slide. You come and orient these tubes. Orient these tubes on a microscope slide. Remember, one end is sealed, and the plasticine at the one end will enable us to attach these tubes at the slide. Then we come and examine at where there is a buffer coat. So this is where there is a buffer coat, where we are suspecting trips. So we come and apply a cover slip. On this slide is where there is a clear buffer coat. But before applying a cover slip, make sure the capillary tubes where the buffer coat is well cleaned. After cleaning it well outside and transparent, come and orient them on a slide, put a cover glass or a cover slip, and examine using times 10 and times 40. But in case, and the procedure they tell us, after orienting them, in the middle we put water. So we put here clean water in the middle. So this is water. Then we examine, put a cover slip and examine. But in case you don't want to use the capillary tubes on the microscope, still you can prepare a wet mount. But this wet mount, you shall pick the buffer coat. You first remove the plasma and pick a drop of the buffer coat and examine it. If there is presence of motile trypanosomes, then you go ahead and perform a stained thin or thick film to see the morphology of the trypanosomes. So this is what we call the microhematocrit buffer method. Lastly, we are going to look at another concentration technique, which is miniature ion exchange centrifugation test. And this one, in this test, here we are using an ion exchange resin. We are using an ion exchange resin, whereby after collecting blood or aspirate or CSF, but majorly we use the blood. So what happens, we use a thin layer, a column, and this column we are using, it contains what we call a cellulose, and the cellulose we use is diethyl aminodiethyl. We call it DEAE. Cellulose is used in the column. So what we do, this DEAE column contains the phosphate buffer, the sodium phosphate buffer. It also contains positively charged substances. And because it is positively charged, we know that blood cells are negatively charged than the trypanosomes. So when we elute blood, if we bring blood to pass through the column, this blood contains the cells which are negatively charged, and as the blood passes through, the negatively charged cells, they are adsorbed on the sides of the column. Because the column, we said, is positively charged, and because it is positively charged, it is going to attract the negatively charged cells. So the negatively charged cells are attracted, whereas the trypanosomes, which might be present, if this is a trypanosome present, for it is less negatively charged, so it passes through. So the trypomastigote, for them, they are eluted and they are found in the eluent. They are eluted. Why? They are less negatively charged compared to the cells. So in the eluent, we are going to find only the trypomastigotes when we have removed the cells. So after getting this eluent, we can go further and we centrifuge to obtain the deposit or the sediment. Then this one, we can apply it on a microscope slide. And we put a cover slip and we examine using times 10 and times 40. And then if it is the trypomastigotes are present, we go ahead and use a stained thin film, a stained thick or thin film to study for the morphology of these cells. So the principle behind miniature ion exchange, why do we call it ion exchange? We are seeing the positively charged column exchanging or attracting the negatively charged red blood cells and white blood cells, allowing the trypomastigotes to pass through. So after correcting the trypomastigotes in the eluent, we can go further and centrifuge, obtain a deposit, and examine using a wet mount, and at the same time stain to study the morphology. That is what we can see. And lastly, we saw ourselves collecting CSF, that is cerebrospinal fluid. Apart from looking for the presence of trypomastigotes, we can also perform the cell count, especially the white BC cell count, and it is always elevated. We can also look for the presence of moat cells, or we can also go ahead and we do also protein, and also protein levels are always high. Then on blood, apart from thick film, direct wet mount, and CBC, we can also perform ESR, and ESR is always raised. ESR is erythrocytic sedimentation rate, which is raised in people with African trypanosomes. So this is what we can discuss with you, both the detection method and the concentration techniques used in diagnosis of African trypanosomiasis. Thank you so much. Hope you have learned a lot in this session. Please remember to subscribe if you haven't, like, and please share to many others so that we may continue to make more videos of such a kind. May God bless you.